Hello, everybody. So I'm just gonna kind of go over our most recent assignment here on Deletion Mutants. I've kind of broken it up into 6 problems: 4 fill in the blank, 4 multiple choice, 5 multiple choice, 6 fill in the blank, 8 multiple choice, 10 fill in the blank. Those that are fill in the blank are English words, otherwise they are random sequences. And as of right now, the multiple choice ones, I always give you the first letter on accident. Um, and otherwise. So there's only one of them where we won't know the first letter really. So let's give the assignment a start. So and now if you want to read through all the steps here, that's basically what I'm going to be following. Um, but I'm going to be doing it uh, a little bit faster, perhaps. So the first thing is we have 4 genes. So on my sheet of scratch paper, I want to create 4 blanks to write down the gene letters. Okay. And first of all, it tells us the first gene at the start of the chromosome is gene T. So I can write down T in the first spot, uh, like a little bit thicker line there. So our first letter is T. Now, how do we determine the other 2 letters? Well, one thing is that we know deletion 2 covers P and S, for example. So we know those 2 are P and S. We do know that this one is gene T because it is told to us. So I guess if we're following my recommended things, if we want to determine gene 2, we need to define a deletion mutant that contains everything except gene 2 and one that does contain gene 2. Um, so deletion 1 and deletion 2 are kind of perfect examples. O, P, and S, P, and S. What's the difference between these two? O. So that means that O better go here. Further, we can also look at the deletion 3 and we can see it's OPT so that we know O and P are either here or here. Right. But from the red, we determine that O is here. And now we have P and S. We know this one's O and P, but we figured out this one's O. So this one, it's TOP. So that one's P. This one means that one has to be S. So it's TOPS. And as step 5 says, let's go through and make sure it all makes sense. So, all right. So I'm going to just go through and write this one be TOP. This one would be P and S, right? And this would be O, P, and S based on the order of the genes. And so if we look at the deletions, deletion 1 contains O, P, S. This one 2 is P, S. These are written in alphabetical order. So this is O, P, T, right? And so our answer tops makes sense. And so we enter that in the blank. And then now we can move on to the next question. Now, this one is multiple choice. I intended to not give you the first letter, but all the choices here have the letter G in them. So we can immediately assume I'm going to go with maybe a pink color. This is gene G based on the answer. Likewise, I'm going to create my 4 blanks. And write in G as our first letter. All right, now if we want to figure out gene 2, it's going to be a little hard because all of our deletions contain gene 2. Only one of our deletions contains gene 4. And only, excuse me, all of our deletions except for one contain gene 3. But since we know deletion 3 is G and J, and this one's G, that means that one has to be J. Um, right? And deletion 1 is G, J, and T. So that one tells us that's T. And this one's J, Q, and T. So the last one has to be Q. And you'll note that the deletion 2 is the only one that contains the letter Q. You know, it's the only one that contains gene 4 as well. So our answer is G, J, T, Q. And so I'm choosing answer B. All right. Now we're moving to a 5-letter blank. This one is multiple choice. The first letter is always B, and the second letter is either A or S. So for that, we can put in the letter B here. And I'm going to create 5 blanks. And I can write the letter B. Again, you can't write on the screen, but you can use scratch paper. All right, we know that gene 2 is A or S, but we don't know which order. We know gene 2 is A, K, Q. So, yeah, I mean, there's a number of different ways to do this. Maybe I should be more systematic with it, but this is A, K, Q, right? This one's A or S. This one's A or Q. It really, that just means that one is A, right? And then none of these are A. So, right. Because this is AKQ or that one's AS, so that one has to be A. And because this is A or S, that one has to be S. Now we know these two have to be K or Q. So if we want to determine gene 5, it's good to find one that contains gene 5 and one that does not contain gene 5. So, so for example, gene deletions 2 and 3, one contains gene 5, one does not. Comparing 2 and 3. They both have A. They both have Q. Deletion 2 has a plus K. And deletion 3 has a plus S. So if deletion 3, that means that one has S and then this one has K. So, you know, the end is K. And then we have A and Q overlapping here. So, I mean, I feel like I'm going a little too fast for a number of students, but hopefully watching me do it will help put the pieces together. BASQK is the one that we decided upon. So I think this one's open-ended. So apologize for the lowercase. I'll get that fixed next year. Do we know the first letter? No, we do not know the first letter of our sequence. So that is a little bit more of a challenge. So 1, 2, 1, 2, 1, 2. Sometimes if I have a long one, I like to put like groups together. So I have 3 and 3. For example, deletion 2 is ALS. That tells me that I don't know what the first ones are, but the last ones are ALS in some order. And oftentimes with my notation, I would use parentheses. So those 3 are ALS. And if we compare deletions 2 and deletion 4, what's the difference between those? ALS and ALST. Right. So we know those 3 are ALS, and these 4 are ALST. What's the difference between those two? The letter T, which immediately tells us that gene 3 is T. So if you're not too concerned about doing these in order, you can do it that way. Another way to figure out, let's say we want to figure out the first one. We can compare deletion 1 and deletion 5 because deletion 1 has the gene next to deletion gene 1 and deletion 5 has the name, but then everything else is the same, right? So the difference between those two deletions is going to be that blank right there. So deletion 1 is AELT. Deletion 5 is AELPT. So the difference is P. That tells us the first letter is P. I don't know if that was too fast for some, but um, that's kind of a strategy. You can, if you want to just kind of pick them out of order, you can do that too. Let's say I want to know what gene 5 is. All right. Well, all of these have gene 5 except deletion 3. So we can look to see which one's missing from deletion 3. Fortunately, we have deletion 5 and we can compare deletion 5 and deletion 3. What's the difference between these two? That one, right? So whatever one deletion 5 has, the deletion 3 does not, is the letter we are looking for. So deletion 3 is A, E, P, and T. A, E, P, and T. The extra one is L. So that tells us that gene 5, 1, 2, 3, 4, 5 is L. So I'm just kind of doing different strategies. Normally, I just kind of take what's given to me. Like we know the blast 3 is ALS. Um, so is gene 4A or is gene 4S? I look at deletion 1. Deletion 1 does not contain S, but it does contain LA, right? So that must mean that A is here. I actually wrote them in the right order, ALS. Now, we can be strategic and try and determine the last one, or we can know that there are 5 different letters. And so whatever letter is missing, we can add there. Maybe you figured that out, but so we're interested in this one right here. Maybe we can compare deletion 1 and deletion 4, right? So we know that one's going to be S, but whatever one is present in 1 and not in 4 would be our answer for gene 2. So 4 is ALST, right? And we can get rid of the A. So ALT, whichever one. So AE is the other one. So we can put that in there. And so what is our word? Petals, like those things you find on a flower. So we can type in petals into our blank. So, all right, next one here should be multiple choice. It's a little bit longer. So we have EFW, but they always start with E, so we can immediately do that. But we do have 8 genes we're dealing with. I don't know the best way to do it. Break it up in 4. All right, and we know the first letter is E based on the choices that we have. Likewise, most likely that second letter is F, but it could be W. So, all right, so let's take a look here. So if we know that first letter is E, I can just put that in there. Um, and no, so if we're just kind of systematically working over, looking at deletion 1, we know that it's EFW, which is the same thing we kind of learned from our things, our multiple choice answers. So I don't know the order, but I can put them in parentheses as FW. Sorry for the bad handwriting. We can figure out the order later. So what's a good one to look at? So if we look at deletion 6, we know that those are going to be FW. So FW, and the next ones are S and Z. So I don't know the order of those two, but I do know those are S and Z. So gene 4 and 5 are S and Z. These two are F and W. Now, is there one that will help us determine the F and W order? We need a deletion mutant that has one but not the other. Deletion 5 is the one to look at. So deletion 5 will contain gene 3, but not gene 2. So if if deletion number 5 contains a W, then we know W is gene 3. If deletion 5 contains an F, then we know F is gene 3. So let's look at gene 5. It says, oh, there it is. It's a W. So that tells us that this gene is W. So that's EFW, and that's W. We know it will contain S and Z, but we don't know the order. And then what other gene here what it contains? So it's WSZ, and then everything, whatever else it contains, is going to be gene 6, right? What else does it contain? D. So it automatically tells us that gene 6 is D. So if you can be efficient with these, more so than systematic. Oh, I deleted the blanks. That's not as much fun. F, W. We don't know the order of S and Z, and then we have what? 3 letters, 2 letters after D. Because we do know this one is D, because that's what we learned from deletion 5. Now, if we want to know the order of S and Z, we need to find a deletion that contains one and not the other. So again, if deletion 7 contains Z, that was the second one. If deletion 7 contains S, we know that's the case. And deletion 7 does contain S. So that tells us that S is second. So this is Z and this is S. And I meant to delete that extra blank because I had too many. So that's the thing with scratch paper. You don't accidentally erase things. All right. So now we have 2 more letters to determine. Which 2 letters do we need to know? Well, what 2 letters do we have left? We have a C and we have a G. So it's a C and a G are either just gene 7 and 8. So we need to find deletion that has one and not the other. Deletion 2. Does it have C? Yes. Does it have G? No. So that means it's C, G. So that's our answer. E, F, W, Z, S, D, C, G. C. Okay, so E, F, W, Z, S, D, C, G. I got to D there. So, all right, the last one, the super challenge one, I guess. We have 10 genes. It's a lot of genes to work with. I don't know. The best way to break it up is 3, 3, and 1. I do tell students that the first one is M. So maybe you could start off with M. Well, it'll be different between, and then 3 blanks, a little bit more, and then 3 blanks, a little bit more, and then 3 blanks, and that might be a good way to solve it. For example, here, deletion 4 are the last 3, E, N, and U. So I can just go, well, these are E, N, and U. I don't know the order. If I want to, I like looking at the smaller deletions first because those kind of less letters to work with, right? So this one's R, T, and U. What's the overlap between those two? It's the U, right? And so I know that these 2 are R and T. I don't know the order. And now I know that the last 2 are E and N. So I'll write that as an E and N. And so if I don't know the order of E and N and their genes 9 and 10, I can look at deletion that contains one and not the other. And deletion 9 fits that one. Which one does it contain? It contains N. So that tells me that N is there, and then E is the one on the end. So now okay, so this is genes 6 and 7 here. Oh, 6, 7. Okay, my kids program me to do that. Sorry, I got to recover here. All right, so we don't know the order of genes 6 and 7. So we need to find a deletion mutant that contains one and not the other. So deletion 1 fits the bill. Doesn't contain R or J or T. Sorry, I know it's a sloppy T. It contains R. So that means gene 6 is R and gene 7 is T. All right, we're getting somewhere. Remember, this is 10 letters, but it needs to form a word, and I think I already got it because knowledge of the English language. We're ending in tune. It starts with an M. What letters do we have left? I have O F I S. Is that right? Fios. Anybody get fiber installed in their house. They have Fios left. Okay. Um, and we have spots to fill 2, 3, 4, and 5. Okay. We don't know. We know Fios is there, but we don't really know the order of those. Um, so we could look at either one of these, and whichever one of the four letters is in deletions 8 and 9. That one would be gene 5, and I'm seeing the letter O. So that means that this one's O. So now we have Orchin. All right. Now we can look at G deletion 7. Um, which one of FIS is present in gene deletion 7? That will be gene 4. I see F ORTU. ORTU. So that means that F is here. All right. You kind of got to take the mutants, the deletion mutants you have. In our case here, it's almost easier to work from right to left, but maybe the deletion mutants that's in your problem might be better to work from right. Sorry, yeah, from right to left, or we're working from left to right, which is kind of backwards. So the two letters we have left are I and S, and they go in genes 2 and 3. So we need to find a deletion mutant that contains one of the two. In this case here, it's deletion 6. It does not contain gene 2, but it does contain gene 3. So if it contains an I, then I goes there. If it contains an S, then S goes in the gene 3 slot. And it does contain an S, so that means that it is S. And then we have an I, and our word that we solved is misfortune. Misfortune. Is that what you had on your books? That is a word. So we can type that in the blank, misfortune. I do allow students to put commas in. It's sometimes it can be working against you. It's better to just kind of type the word cleanly, I think. So misfortune is our word right there, and we can hit submit, and we are done, and we can see whether or not Dr. Voss got them all correct. So question 1 was correct. Question 2 is correct. I got number 3 wrong. I'll go back and look at that one. Question 4 is correct. Question 5 is correct. Question 6 is correct. So what did I do wrong? We didn't go back and check our answers. That's one of the problems. So I wrote BASQK, which may not have been the one I wrote down. BASQK. All right, so if we have BASQK, then deletion 4 should be BASQ, and I'm probably going to guess ABQS. Okay, so that checks out. Deletion 2 should be SQK. Deletion 2 is AKQK. So I think I swapped those two. So the answer is actually BSAQK. Sorry. So gene 2 is SA. So the only one that's going to tell me the order of A and S is that one, right? Because that's the only one that contains 2 and 3. And because deletion 2 contains A, not S, it's BSAQK, not BASQK. So that's what I did wrong there. So hopefully it was just an entry problem and not a systematic problem that I had earlier, but that's why it's good to go back and check our answers. So at least I did the other ones right. But it was misfortunate that I got that one wrong. So anyway, thank you for watching. If there's any more videos or something that's not quite clear, feel free to reach out. You can use the comments. Students know how to get to a hold of me directly. And thanks for watching.